So, it's my pleasure to present you the last speaker of the day is Dr. Ana Abril from University of Santiago de Compostela and SIC in Spain. And she continued the fil rouge of this second part regarding the Listeria and production of virulence factors and antimicrobials. And so thank you for coming, and please.
Good morning to everyone. Thank you, Paola, for the introduction. And of course, thank you for inviting me to this conference. I would like to talk to you about proteomic characterization of antibiotic resistance in Listeria and production of antimicrobial and virulence factors. I want to show you how in this study we characterize different Listeria strains that are pathogens and were isolated from food by the identification of bacteria peptides that correspond to different virulence factors by liquid chromatography, electrospray ionization, tandem mass spectrometry. This study allowed us to determine the strain variation and the pathogenic serotypes of the different strains that we analyzed. Some Listeria species are important human and animal pathogens, and Listeria can colonize a wide range of different environments. Listeria can be transmitted through contaminated food, such as milk, poultry, and meat. And as the professor talked us before, Listeria contamination is common in food industry, forming biofilm that can survive for months and even for years. Some Listeria strains have multidrug resistances, and these resistances are now increasing, as while antibiotic-resistant bacteria present in food are initially saprophytic or commensal, and the resistant genes are transferred to other microorganisms in food. This is why an effective therapy is needed, and also the identification of these strains that are pathogens, as well as determine its virulence factors, such as antibiotic resistance and sensitivities. In this slide, we can see how Listeria colonize the gut barrier epithelium with the attachment of the Listeria to the receptors by the internalin A and B, then with the invasion, and with escape from the host phagocytic vacuole by the pore-forming toxin Listeriolysin O and the phospholipase A and B. Then Listeria evades host immunity by different mechanisms, including again the toxin Listeriolysin O and flagellin and internalin C. The formation of the actin tail is provided by the actin assembly inducing protein that allow the cell-to-cell spread, and then in a new cell, again, Listeria can escape from the host phagocytic vacuole with the final release of the organism. In this table, we can see all strains that we use in this study. 8 of the strains were obtained from the Spanish type culture collection, and a strain 4, that is a Listeria monocytogenes species, were obtained from the collection of the Department of Microbiology from Santiago de Compostela University. All the strains were characterized by Malditov mass spectrometry and also by the 16S RNA sequencing to certify the species they belong. As a resume of the material and methods, we start with the peptide sample preparation, following by the liquid chromatography tandem mass analysis, and the processing and bioinformatic analysis with the final protein identification. For the peptide sample preparation, the Listeria strains were cultured in braid haired infusion and then were transferred to plate count agar, both with the same conditions of 31 Celsius degrees for 24 hours, and all samples were done in triplicated. Then the protein extraction were performed, and these proteins were denatured, reduced, alkylated, followed by the tryptic digestion, and the resulting peptides were desalted and purified in a C18 microspin column. Then for the liquid chromatography tandem mass spectrometry analysis, we used a UHPLC Proxion ECNLC2 nanoflow system from Thermo Fisher with a reverse phase column and a per column aquacore XL-C18, both from Thermo Fisher. For each sample, we use 1 to 2 micrograms per run. And these UHPLC were coupled to a LTQ Orbitrap XL mass spectrometer from Thermo Fisher in positive mood, followed by 10 data-dependent collision-induced dissociation, tandem mass scan using an isolation width of 3 mu, and a normalized collision of 35% of energy. For the processing and bioinformatic analysis, we compare our data, the peptides that we obtain, against Listeria CI, Uniprot, TIMI-BL, what have 267,496 protein sequence entries, and we use the sequest program for protein identification. Percolator algorithm was used to validate the statistical analysis, keeping a false discovery rate less than 1%. To identify the virulence factors among our data, we compare the proteins against virulence factors of pathogenic bacteria database, and we also review different previously reported data and articles. And finally, we obtained 2,990 non-redundant peptides, which belong to 2,727 annotated proteins from Listeria. Among these proteins, we have found 395 peptides of proteins that play a direct role in Listeria pathogenicity. And among these proteins, we found toxins, antimicrobial compounds, antibiotic-resistant peptides, and peptides to other resistance to other toxic substances, also ABC and other transporters that are related to virulence factors, proteins involved in colonization and immune evasion factors, and finally, alternative virulence factors. In this table, we can see the total virulence factors for each strain that we analyze. In case of strain 2, that is a Listeria serengeri species, we have found 116 peptides, and in case of strain 4, that is a Listeria monocytogenes species, we have found 73 peptides. These were the strains with a higher number of peptides related to virulence factors, what suggests that they could be the most pathogenic strains. However, strain 2 were described previously as a virulent. And strain 3, that is a Listeria innocua species, we have found 55 peptides related to virulence and was described as group 1. And the remaining strains that we found less peptides were described as group 1 and group 2 of pathogenicity. We have identified 82 peptides involved in bacterial resistance to antibiotics and other toxic substances. Among them, 49 are associated with antibiotic resistance, and the remaining 33 are related to other tolerances. And in this table, you can see all the proteins that we have identified related to this. And here, some examples of antibiotic resistance is an aminoglycosid N3 acetyltransferase. That is an enzyme that can provide resistance against a variety of antibiotics. Another example, the MIR-R, that is involved in resistance against several antibiotics too. Other example, the M56 peptidase, that is a metallopeptidase that is involved in beta-lactamase transduction. Other example, TR proteins that play a role in the regulation of genes encoding small molecule exporters and antibiotic resistance. And the IDEL that plays an important role in antimicrobial peptide resistance. All strains here we have identified different peptides related to antibiotic resistance, except strain 9, that is a Listeria jovanovis species. Related to additional resistance and tolerances, we have identified different head shock protein chaperones, also a TLA protein that confers telluride resistance. We also identified MER, a metal ion sensing regulator that creates a mercury resistant phenotype. A SUG, that is a small multidrug resistant protein. PEDR, that is an environmental sensor that acts in the phenolic acid stress response. We also identified 2 peptides of antibacterial proteins, among them a bacteriocin strain 4, that is a Listeria selengeri species, and antibiotic biosynthesis monocygenase that in strain 9, that is a Listeria monocetogenes species. Moreover, we identified 12 peptides that are involved in bacteria toxicity. Among these toxins, the toxin set that stabilize resistant plasmid in major human pathogens, the LHG domain containing protein that is present in the N-terminal region of group of polymorphic toxin proteins, the HICA toxins that function as a translation-independent mRNA interferase, and we also identified 2 type 2 toxin-antitoxin system, between them the really toxin that are mRNA interferase and PA toxin that inhibit urease activity. On the other hand, the PIMK MAZ-theta toxins inhibit cell division, resulting in cell death. We have identified 131 peptides that were identified as belonging to proteins that play a role in Listeria colonization and immune evasion. As an example, we identified an autoinducer 2 transporter, which is involved in quorum sensing signal and biofilm formation. We have determined a huge amount of peptidase and proteases, as examples the CLP protease that hydrolyze the protein controlling the modulation of virulence factors. Another example, the insulinase that cleaves small proteins and peptidase during bacterial invasion. We also identified bacterial surface proteins that play a role in the attachment of Listeria to the receptor. As examples, different internalins and other adhesion proteins. We have identified 2 proteins of competent system proteins, which are involved in the escape the host phagosome and research the cytoplasm. A peptidoglycan O-acetyl transferase that is involved in avoiding eukaryotic lysosome recognition. Different lysosome proteins that are autolysins involved in bacteria adherence to eukaryotic cells. We also determine different transcriptional regulators, which regulates the expression of different virulent genes. The superoxidimutase enzymes, which catalyze superoxide radicals produced by host macrophages and neutrophils. Also, we identify different proteins of secretion systems. These systems facilitate the secretion of extracellular proteins across the cytoplasmatic membrane. And finally, we have identified different proteins that are involved in the biosynthesis of thiotic acid and lipothiotic acid proteins. These proteins are involved in inflammation reactions in the host. We also determined 113 peptides of ABC transporters that are involved in virulence, such as multidrug transporters, antibiotic transporters such as pectrobactin and others, and ion transporters. And we have identified other 12 peptides that belongs to other transporters that are not ABC transporters, and again multidrug transporters and ion transporters. Finally, related to other bacterial virulence factors, we have identified 43 peptides, and among these proteins, we identified proteins involved in the acquisition of mobile elements such as integrases, recombinases, and transposons, and also specific plasmid proteins. And as a resume, the analysis here that we report involved an in-depth study of the antimicrobial and virulence factors and present in 9 isolated Listeria strains using shotgun proteomic tools with the proteomic isolation, the peptide separation, and the database search with the final protein identification. And the analysis identified 2,919 non-redundant peptides, of which 395 represent proteins that are in Listeria virulence factors, such as including toxins, antibiotic resistance peptides, and other proteins involved in colonization and immunization, among others that I told you before. And as conclusions, we can say that strain 2 and strain 4 are the Listeria strains containing the highest number of virulence-related peptides identified, indicating that these represent the most pathogenic bacteria as compared with the other strains analyzed. The methodology allowed determination of specific virulence factors as displayed by the bacteria in situ within a particular environment, while laboratory conditions does not always reflect the characteristics displayed by a particular strain in a different environment. This approach may contribute in the development of a rapid and accurate identification of pathogenic bacteria, including Listeria species, which is an essential issue to maintain a good quality food chain. The proteomic repository obtained here can be the base for further research into pathogenic Listeria species and facilitate the development of novel therapeutics for these pathogens. The precise proteomic method implemented in the present study represents a useful step for further analysis and pathogenic bacteria, and it offers considerable advantages over traditional approaches in terms of speed and reliability, without the need for full genomic sequencing analysis. And finally, I would like to thank to every collaborator in this study, and also thank you for your attention, and again for inviting me to this conference.
Thank you very much.